logged on. So I'm very excited today to have a talk on something that's a little bit different. So Physcomitrella, we don't, so this is a moss, we don't have very many. I was really intrigued by this paper when it came through because it gives us a glimpse. It's looking at chitin signaling and calcium responses in a moss. So very, very ancient, going far back in the evolution of plants, which really tells us that this interaction between fungi and plants has been going on for a long time and might give us a sense of how that has evolved. I'm personally very interested because I study symbiosis and chitin signals and lipocyto-oligosaccharide signals trigger calcium spiking. And so I'm really interested in how this signaling mechanism has been co-opted for nodulation, but also to understand that you need to know the basis, the background. So that really caught my eye, and it hasn't just caught my eye. It's been picked up by 4 news sites, so definitely there's a lot of interest in the paper. And I believe that Ashley is going to type the link into the chat, so anybody who wants to follow, and this is also a good place for me to remind you that if you want to follow up on any of the things that from the seminar today, you'll be able to look at the data, at the figures, at the methods, and really dive in a little deeper. So to my co-host today is Dong Wang, who is from UMass Amherst. Dong Wang is a member of the MPMI editorial board, and he's also one of the guest editors of our special focus issue on how do plants simultaneously interact with beneficial and pathogenic microorganisms. Other guest editors are me, Olga Zucchero, and Kay Jones. And the deadline for that is November 14th. If you have any questions about whether a paper is appropriate, you can email any one of us. Our speaker today is Giulia Galotto, who is the first author of this interesting Physcomitrella calcium signaling paper. And this is part of Giulia's graduate work with Luis Vidali at Worcester Polytechnic Institute. And she is just finishing up. So she is going to be wrapping up her PhD literally this week. So timing is very appropriate. Okay, so I am going to turn it over to Giulia now, who is going to share her screen and tell you about the paper. At the end, if you have questions, please type them in the Q&A box that you'll see at the bottom. And we will, Dong Wang and I will take turns reading them, and Giulia will answer, and we should be able to get through quite a few of them. So looking forward to that. Go ahead, Giulia, and share your screen. And then if, once she does, if you click speaker view, I think that makes the rest of us go away so that you can focus just on the seminar. So.
Okay. So you should see my screen.
We do. Okay. All set.
Okay.
Thank you, Giulia.
Thank you, Jen, for this introduction, and thank you for all of you to come here today to listen to my talk. So the title of my talk is Chitin Triggers Calcium-Mediated Immune Response in the Plant Model Physcomitrella patens. So this is the outline of today's talk. I'll first talk about the background, about plant immune response and plant pattern recognition receptors. And then I'll talk about calcium as an hallmark of plant immune response. In the results part, I'll talk about mainly about the results of the paper that we recently published in MPMI. And then I'll present some data that are relevant to this story, but that were published as a part of a different paper recently from our lab. Just get the pointer. And then we'll conclude discussing the future directions. So plant growth and survival are threatened by a variety of biotic and abiotic stresses. And for plants to survive to such stresses, they evolved an immune system. This is a schematic of the immune system at the cell level. And as you can see, the defense system starts at the plasma membrane, where plants exhibit PRR receptors, pattern recognition receptors, which interact with pathogens or microbe-associated molecular patterns, PAMPs or MAPs. When a PRR interacts with a PAMP, the plant starts the PAMP trigger immunity, or PTI. And this type of immunity involves ROS production, calcium signaling, calcium increase, MAP kinase phosphorylation, and hormones production. And all these events culminate in the transcription of immunity-related genes. In parallel, pathogens evolve the mechanism to circumvent PTI and eventually to stop it, and they do so by secreting effector proteins in the cytoplasm of the cell. The plant cell can actually sense this effector protein, and LR protein can bind to effector proteins, and this event triggers the effector trigger immunity, ETI. ETI is more dramatic than PTI, and it can culminate in the hypersensitive response and eventually also in plant cell death. So for our talk today, we're going to focus on PTI and specifically on the immune response of Physcomitrella patens plants to chitin, to fungal chitin. So the first thing I'll do is I'll present the pattern recognition receptors for fungal chitin in plants. So PRR for chitin have been mainly studied in rice and in Arabidopsis. And rice is actually the first plant in which the receptor for chitin has been identified, and this is the protein sebib. So two molecules of sebib binds to a chitin, and upon this binding interaction, they seem to recruit SIRC1, and they form an hetero tetramer. In the case of Arabidopsis, there are two proteins that can bind to chitin. One is SIRC1 and one is LIC5. But LIC5 actually binds to chitin with much more affinity. So SIRC1 can act as a receptor for chitin, but the main receptor is LIC5. And this receptor works in a similar way to the rice one. So LIC5 binds to chitin, and upon binding of LIC5 to chitin, they recruit SIRC1, and then SIRC1 will be activated and then simulate a downstream signaling. In the case of Physcomitrella patens, until recently, we only knew that SIRC1 was important for chitin sensing, but today we also know that Physcomitrella patens as Arabidopsis as LIC5 that mediate the chitin sensing, and I will present so these data come from our lab, and I'll present some of these data today. So after a PRR recognizes chitin or other pathogens, the plant activate a downstream response. And the message of this slide is that the downstream response of vascular plants and mosses are similar. So they involve a calcium, cytosolic calcium increase, MAP kinase phosphorylation, ROS production, hormones production, activation on transcription-related genes that then activate downstream signaling, and other events such as production of callose and pre-lignin compounds and so on. So for the talk today, we're going to focus on the cytoplasmic calcium increase that happens after a pathogen, after a plant sends a pathogen, because this is one of the first events that occur. So increase of cytosolic calcium is a hallmark of plant immune response to both biotic and abiotic stresses, and this has been very well characterized in Arabidopsis. So on the left of the slide, we can see Arabidopsis seedlings that are expressing equine, which is a calcium sensor. And here the seedlings have been treated with flagellin, which is a bacterial protein, with PEP1, which is actually a plant protein that amplifies the response, and from sodium chloride. So we can see that in Arabidopsis, both a biotic and abiotic stress trigger a cytosolic calcium increase. On the right side, we can see the case of more adult Arabidopsis plants, and this is the case of an herbivore attack, and these plants are expressing the sensor GCAMP. Here is the site of herbivore attack, and you can see that upon this attack, the cell has an increase of calcium that then is propagated to the rest of the plant. So in the case of Physcomitrella patens, we know that an abiotic stress can trigger a similar calcium response. This is work that has been done by Storti et al. And they show that when we treat Physcomitrella patens plant with 500 millimolar mannitol, there is a burst of calcium at the base of the gamedophore that then will propagate to the whole gamedophore. And these are Physcomitrella patens plant that express the FRET-based sensor chameleon. So whether we know that an abiotic stress trigger a cytosolic calcium increase in Physcomitrella patens, we still don't know if a biotic stress also triggers a cytosolic calcium increase in Physcomitrella patens. And this is the biological question that our paper addressed. So before I answer this question, I want to introduce the model system that our lab uses and that we also used in this paper. And this is the MOS model Physcomitrella patens, which is a bryophyte. It is a very amenable organism to work with. It can perform homologous recombination with high efficiency. It can perform transient RNAi. This MOS can be propagated vegetatively, and that means that you can collect the tissue, you can ground it, and then you can plate it, and the tissue will just grow again. And then it has a predominantly haploid life cycle. This life cycle is depicted here. So the plant originates from a spore, from a haploid spore, and from this spore it forms a peritoneal tissue. This peritoneal tissue is formed by filamentous cells that grow by tip growth, and this apical cell grow and then divide, and after the third cell division it forms a branch, a lateral branch. This branch will proliferate into a bud that will then differentiate into a gametophyte. And after fertilization on top of the gametophyte, it's formed a sporophyte, which is the only diploid stage of this plant. And then meiosis inside of the sporophyte will produce the haploid spores. So in our lab, we work on the juvenile stage of the plant, which is this peritoneal tissue. It is a single cell thick layer tissue that looks like this. And in this tissue, there are only two cell types. And these cell types are chloronyma cells and colonyma cells. The chloronyma, they grow slower than colonyma. They have higher number of chloroplasts and they have perpendicular wall. The colonyma grow faster. They have oblique walls and they have fewer chloroplasts. So here I have a video that shows how this plant grow, because this is a very peculiar mechanism. So I want to show it to you. And what I also want to show you is that we can image a whole 1 week old plant with a 10x lens in the field of view of the microscope. This is particularly good when we want to observe any mechanism at the whole organism level. So this is a about 3-day-old Physcomitrella plant in which the cell wall has been stained with chalcofluor. So you can see the tip growth mechanism. The cells are growing from the apex, and you can see that they're dividing here, and then they will form a lateral branch here, and then a lateral branch here. So you can see at the end of this video, the plant is about a week old, and we can image the whole plant in this way. Okay, so now we can go back to our biological question of today. So does a biotic stress trigger a cytosolic calcium increase in Physcomitrella patens? So to answer this question, we need a fluorescent tool and a fungal elicitor. So as a fluorescent tool, we use a Physcomitrella patens line that expresses the calcium sensor GCA that you can see here on the right. This is formed by a circularly permutated enhanced GFP that is bound to calmodulin and M13. And upon calcium binding to calmodulin, there is a conformational change that will render the protein fluorescent. Since actin is very important for the growth of Physcomitrella patens, we also transform these plants with the sensor for actin, which is LifeAct Ruby, so that we can look at the actin cytoskeleton during a fungal infection. To simulate the fungal infection, we use a variety of ways. We use chitin oligosaccharides, which is a mixture of oligosaccharides of different length of N-acetyl glucosamine units. So it's really an unknown mixture. Then we have chitotriose, which is formed by which is a polymer of 3 N-acetylglucosamine units. And then chitohexose, which is 6 N-acetylglucosamine units. So now we can use our plant that expresses the calcium sensor, and we can treat it with chitin, and we can observe the calcium signaling. So here I have video that shows this treatment. And before I play it, I just want to point out that this is the control plant treated with water, and this is the plant treated with 1 micromolar chitohexose and 100 micromolar chitohexose. So as you can see, the control plant exhibits just a few calcium spikes at the tip of the cell, the cells, but the plants treated with the chitohexose have a big calcium wave travels across the whole plant. And we can see this in the 1 micromolar case, and we can see an even more dramatic effect in the 100 micromolar case. So when we, so I'm gonna show you how we quantify this data. So we decided that to measure the intensity of the signal, we calculated the mean fluorescence of the calcium signal. And then to get a sense of the amplitude of the signal, we calculated the standard deviation of the fluorescent signal. So I'm gonna show you these graphs now. So here on the left, we can see the mean fluorescence, and on the right, the standard deviation. And in red is the chitohexose treated plant, the one that I just showed you, and then in blue, the chitotriose. And as you can see, plants treated with chitohexose have a higher fluorescent intensity when we treat them with, the more chitotriose we use. And similarly, there is a higher standard deviation in plants that are treated with higher concentration of chitohexose. On the other end, when we use the chitotriose, we have a very minimal response, and we only basically have a very low response when we treat them with the highest concentration of chitotriose. And this is interesting because this shows that there is we need a minimal number of N-acetylglucosamine units for the plant to actually trigger a response. So we further analyzed this data, and we decided to quantify the calcium peaks that we observed. And to do that, we used a MATLAB algorithm that was developed by Kat Sherman in our lab. And so briefly how this works is that there is a 3x3 pixel grid that is the square. And this calculates the average fluorescence of the pixels inside of that square. And the square travels across the plant and calculates the mean fluorescence, and then across each plant in the time series. And then we calculated the fluorescence of the whole time series and the standard deviation of that. And a signal qualifies as a peak when it is greater than a fraction of the standard deviation of the whole series. This is an example of the fluorescence detected by one of these squares across the time series. So after we calculated all the peaks, we plot the number of peaks and the peak frequency in both the chitohexose and the chitotriose case. So as you can see in both cases, there's a higher number of peaks and higher peak frequency when plants are treated with higher concentration of chitohexose. And in the case of the chitotriose, we have a very minimal, basically no difference between with the control. Okay. So now that we answer our main biological question, we wanted to study how the chitin affects plant growth and solidity. So we performed a growth assay experiment, and we measured the plant area and the plant solidity. So the area is just the area of the plant, and the plant solidity is a measure of how the plants are branched. So plants that look like this with a lot of branching, they will have solidity values close to 0, while spherical plant without branches that look like this will have solidity values close to 1. So we regenerated single plants, and then we exposed them to higher concentration of chitin oligosaccharides in this case. And then we detected the area and solidity. And as you can see, when we increase the concentration of chitin, the plant area the area is smaller, is progressively smaller, and then the plants are progressively less branched. And also in green, we are staining the cellulose, and you can see that when we increase the concentration of chitin, there is a less homogeneous distribution of the cellulose. So we wanted to understand how is that the chitin has such a dramatic effect on plant growth. And as I mentioned earlier, the actin cytoskeleton is very important for the growth of these cells. So this is a control cell, and you're looking at the actin cytoskeleton, and you can see that a cell that is growing has a very dynamic cortical actin, and it has an accumulation of actin at the tip of the cell. And this actin spot is very important for growth. All the cells that are growing exhibit this actin spot. So to investigate if the growth defects are due to defects in the actin cytoskeleton, we treated these cells with 100 microgram ml of chitin oligosaccharides, and we observed the actin cytoskeleton. So as you can see, about 2 minutes after the treatment, the actin accumulation at the tip is dissipated. So we attribute the defects at the growth at the plant level to defects in the growth in the cell level due to the dissipation of this actin cytoskeleton at the tip, which is essential for growth. So then we measure the apical calcium in these cells, and this is the calcium oscillations of a control cell. The oscillations are periodic, and they are only restricted to the apex, to the apical domain, and the cells exhibit frequent peaks of variable amplitude. When we treat these cells with chitin oligosaccharides, we can see a few big peaks that are not only localized at the tip, but in the whole cell. And then the signal plateaued out, and there are just very little oscillations. Okay, so the last experiment of the paper that I want to show you is we wanted to verify if the calcium is sufficient to trigger the transcription of defense-related genes in Physcomitrella patens. So to do that, we used a calcium ionophore, which is ionomycin, which can artificially increase the concentration of calcium in the cytoplasm. And the genes that we picked are the one that, thanks to Bressendorf et al., we know they are that their transcription is increased upon chitin treatment in Physcomitrella patens. And as you can see from this dark gray bar, calcium alone is sufficient to trigger the increase in transcription of these defense-related genes. So this concludes the first part of the talk, in which we observed that calcium oscillations originate following elicitation with exosaccharides. Chitin oligosaccharides affect plant growth and morphology in a dose-dependent manner. And we saw that chitin oligosaccharides dissipate F-actin at the tip of the cell. We also observed that several defense-related genes are expressed in chitin oligosaccharide-treated plants, and they can also be induced by artificially increasing cytosolic calcium. So these results indicate that Physcomitrella patens' immune response to chitin oligosaccharides is calcium-mediated. So now, I'd like to show you some data that were published in, that are relevant to this story, but they were published in a different publication recently from our lab. So we wanted to identify which one is the receptor for chitin in Physcomitrella patens. And both Arabidopsis and Physcomitrella patens have a cytosolic calcium increase. So we wondered if also in Physcomitrella patens, as in Arabidopsis, Liq5 participate to calcium sensing. So we did a phylogenetic analysis, and we found that Physcomitrella has 3 Liq5 genes that are similar in sequence to the Arabidopsis Liq5. And we designed an experiment in which we RNAi the Liq5 receptor, and we observed if we could see the calcium oscillation. So this to verify if Liq5 is essential for this pathway. So briefly, I want to show you the mechanism of RNAi that we used, because this is a novel RNAi mechanism that our lab developed. and it exploits the APT silencing system. So APT is an enzyme that converts 2-fluoroadenine into cytotoxic nucleotides. So silencing of APT confers resistance to 2FA. So if we do RNAi of APT and we plate plants on medium that contains 2FA, the plant will survive. But if we don't silence APT and we plate plants on 2FA, they will no longer survive. So we combine the RNAi of APT to the RNAi of the LIC5 family, all the 3 genes. And then we plated the plants on 2FA to select for the silencing plants. Then we exposed them to chitin and we repeated the assay, the calcium assay. So these are the data, and this is the montage of the data. And in the first row, there is a wild type that, as you can see, exhibit the big calcium spikes that we saw before. The control of the RNAi also exhibit this big calcium spiking, as you can see. But the plants in which LIC5 has been silenced do not exhibit the calcium spiking. So the plants in which we silence the LIC5 don't sense chitin anymore. So then we quantify the data in the same way that I showed you before, so that we measure the fluorescence, the standard deviation of the fluorescent, and then the number of peaks and the peak frequency. And in blue, there is the control of the RNAi, and in red is the LIC5 RNAi. And you can see that when we do the RNAi of LIC5, the response of calcium is basically nothing. It's basically 0. So, thanks to this data, we can conclude that there is a functional homology between Arabidopsis LIC family and the Physcomitrella patens LIC5 family. So the LIC5 family functions at the level of chitin perception in Physcomitrella patens, as in Arabidopsis. So as for future directions, it would be interesting to answer this question. So in Arabidopsis, it is necessary for chitin to bind into LIC5 for the activation for the heterodimerization and activation of SIRC1. And it would be interesting to verify if this mechanism is also conserved in Physcomitrella patens. Okay, so these are the acknowledgements. So I'd like to thank the lab, mainly Professor Luis Vitali, for his endless patience with me. And then, so Kat is the student who designed the MATLAB script that detects the peak, and she also worked on the RNAi of LIC5. Isidro also worked on the qPCR and also on the designing of the LIC5 RNAi. Rob and Steven worked on the APT system. And then I also like to thank all the other members of Vitali Lab for support, Professor Guerrero, which is a collaborator, and all the funding sources. Also, I'd like to thank you for your attention, and I'll take questions.
Thank you very much, Giulia, for a really interesting talk. Really enjoyed it, and I'm looking forward to seeing what people ask. So just to remind you, please type your questions into the Q&A, and Dong Wang and I will take turns reading. So I'll take the first one. Erbil Gungor asks, where does the calcium emerge from? Is it stored somewhere?
So, so usually in plant cells, the calcium is stored in the ER and in the vacuole. So those are, so that's a possibility. Those are the two organelles in which the calcium is stored, and yeah.
Okay, Don, do you want to read the next one?
Sure. Yeah. The next question is from Mark Knight, and he asks, if you inhibit the calcium increase in response to chitin, I imagine using small molecules, drugs, etc., does that prevent the downstream expression of defense genes that you tested?
So we didn't do that experiment, but I think it would be interesting to look for that, and I anticipate that would be the case. According to our data, I anticipate that would be the case. Yeah.
Okay. I think people are typing them into the chat. I'm going to ask the people move them to the Q&A, but I will read one from the chat. Luis Cardenas asks, the silenced like 5 is no more growth sensitive to higher concentrations of chitin. So is it more growth sensitive?
So I don't. So the LIC5 RNAi is less sensitive to what? Sorry.
Whether its growth is, you were showing the response, the calcium response, and I think he's asking about tip growth.
The growth assay. Oh, so we didn't look into that. So I guess we would have to repeat the growth assay. So we didn't look into that at the tip level. So if you're asking if the actin will be affected.
Yes, I think that's the question.
Well,
not even the actin, but even just really the growth.
I don't think that it would be affected, considering the result that we have.
Mm-hmm. Interesting.
I mean, if LIC is the only receptor, then it would not be affected. If there is some phenotype, then maybe there are some co-receptor. In the case of Arabidopsis, there's also SERC1 that has can detect chitin. So if that would be the case, then we could see maybe it's a mild phenotype, intermediate phenotype.
Yeah. Why don't we stop sharing your slides since we're moving to the Q&A part. I think that will be, perfect.
There's a few questions in the Q&A session also. So the first one is from Nohemi Carreras Villasenor, asking any condition that increase calcium can increase expression of defense genes? Or I guess the question is, if you somehow increase calcium, cytosolic calcium concentration, does that just induces defense genes on its own without elicitation by chitin?
That was the experiment that we did. So we did either elicitation with chitin or just the ionophore. So when we do the experiment with ionomycin, it's only ionomycin. There's no chitin. So when we see the increase of those transcripts, is only due to the artificial increase of calcium.
That's just asking for the confirmation of the result. Yeah.
Mm-hmm. Okay. So the next one, I don't have a name with it. Asks, the chitin treatments you used a relatively high level, so the 100 micromolar, but you also used 1 micromolar. So the chitin treatments you used are relatively high levels. What is physiological? And that's always hard to know.
What is the physiologic
level?
If there was a real fungal infection, what would be the?
Well, maybe how about compared to other studies, right? So in Arabidopsis, levels of chitin.
So for compared to other studies, sometimes it's not easy to do that because we use the actual chitotriiols and chitoexols, but other study, if they use the chitin oligosaccharides, it's a concentration, but we don't have a molarity because that's mixture of unknown. So we would have to calculate from the molecular weight. But so I'm not sure how to answer this.
Yeah, I think it's actually a interesting question, because if you actually had a fungus there, right, there are going to be chitinases that are breaking down the cell wall, releasing little fragments, but it's very hard to know locally, like extremely locally, like right by the hypha or haustorium, how much of these little fragments would be released and what sizes would they be. So it's a difficult question to ask, but it is important to think about, it's a difference in a real fungal infection or interaction with a symbiotic fungus. You're not experiencing the same thing over the whole plant. So it's an important question. Okay.
All right, so next one is from Carlo Peroro. And he points out that he noticed that neither Arabidopsis or Physcomitrella form a bascule mycorrhizal symbiosis, which is widespread in other species. So he asked, if you look at the sequence sequence of the LIC5 proteins in these two species versus other species that do form a vascular mycorrhizal symbiosis, do you see any difference? Because I think it's known that at least in some cases, demonstrating some species that the LIC5 is required for symbiotic fungal interaction as well.
So we didn't look at this, but if there is a functional difference, if there is a different mechanism, it's possible that there is a difference in the, I don't know, in the structure, in the sequence, but maybe there is some difference. I'm not sure.
Yeah, that would be a very interesting alignment to take a to see, right? So
yeah.
Now you have two species that do not form AM symbiosis. Yeah.
Yeah, it's an interesting base to start a more evolutionary thought series. So Juan Carlos Diaz-Vici says, nice talk, Giulia. Have you detected ROS accumulation or changes of the expression level of CAMs or CMLs when the system is activated with chitin?
So we didn't look for it. So we didn't do those experiments. I think it would be interesting to verify that in the future, but we just didn't get around to do it.
Yeah.
Okay, next one from Mikey Lemons. Hi, thanks for a nice talk. Wondering what the KD50 value is for your chitin hexose.
Yeah.
High concentration. Oh, yeah, that's the first one.
So we measured that. I don't. It's like I don't remember exactly. It's like one. I don't remember exactly, but it's close to the Arabidopsis one for chitin, which makes it interesting. I think close to 1. I think it's 0.1 micromolar.
Yeah. And she also had a question, which was already asked about the concentration of the chitin you use, but it's related to something that I have in mind, okay, which is your growth assay was using a different measurement of the chitin concentration. So how does it compare to the calcium oscillation work?
How would that compare to the calcium?
Yeah, calcium oscillation, you're using micromolars, right? Here, and then in the growth inhibition, you're using micrograms per mil. So.
Yeah, because in the case of the growth assay is the chitin oligosaccharides.
That's a different kind.
In the case of, it's an oligosaccharide, so we don't know the molar.
Yeah.
That's
gotcha.
This actually reminds me that some of these specific questions, like about the KD50, reminds me that we have that the link to the paper is posted in the chat. And this is a great opportunity for people who want to look to really zoom into the details, the methods, the supplement. There's a lot of information in there. So I hope some of you will also turn to that. And I think Ashley is typing that into the chat again for those who joined a little later. Okay, I'm going to ask, there's another attendee without a name here. It says, why is calcium spiking always used as signal transduction in plants?
You actually break up a little there. So
sorry. That's what happens when you're in a house where you're not the only person using the internet. Okay. So why is calcium oscillation or spiking always used as signal transduction in plants? I'm not sure it's always, but it's certainly often used.
Why is calcium used in signal transductions in plants?
Yeah.
Okay.
Very philosophical question.
Well, I mean, so why is it used? I mean, there is a lot of proteins that are sensitive to calcium. So for all the calmodulins or all the calcium binding proteins, but that probably it's a mechanism that evolved after the cell decided to use calcium as a sensor. I mean, I don't really know how to answer that.
I think calcium signaling is such a ubiquitous.
Yeah.
I mean, T cells use calcium signaling. It's I think it's probably because it's so easy, you know, calmodulin is so ancient. You got that sensing component, and it's so easy to rapidly increase calcium in the cytoplasm and rapidly decrease it. So you can be very fine-tuned, or maybe it started off not being fine-tuned and now it is. I don't know. It's a sort of a philosophical question there. Okay.
The next one is from Valdir Morera. And excellent talk. And if I understand his question properly, it's asking whether your system for RNAi can be used in other species, for example, in Arabidopsis, knockdown genes as well.
I think yes. I think that yes, it can be used in other plant species. Yes. I don't know the details, so I can remind you to the paper, but yeah, I know that it can be adapted to other plants.
It has been demonstrated that it works in flowering plants as well.
Yeah, but this is something that it's the first time used in Physcomitrella.
Yeah, we developed this.
Yeah, interesting. That is a really interesting kind of selection.
It was used before, but in a knockout. In our case, we do an RNAi, so that was the novel part that we added.
Mm-hmm. So Sonali Roy asks, what is known about the like receptor family in Physcomitrella? Is it possible that there are other receptors that might recognize different lengths of chitin?
I think so. I don't. We could look again at the tree. I don't remember exactly, but there could be other receptor. I don't remember if there are. So there are 4 circ in Physcomitrella, and so it is. And then of these 4, like 2 transcript are more increased upon chitin, and so we know from Bressendorf et al. That circ1 is essential for the signaling transduction, especially for the MAP kinase phosphorylation. So it is possible that circ is also it is a receptor for chitin. I don't know if it's only working together with LIC5, or if it could be also independent receptor, like in Arabidopsis. So, yeah.
So are there other? So I don't recall the phylogeny, but you have the little like 5 group. Were there other clusters? Other like? I mean, I'm trying to remember how much it's diversified in Physcomitrella. I think there were a lot.
Of LIC5? Only.
Or what? No, there are 3 LIC5, but other LIC, like for example, in Medicago, there are many different ones, not just LIC5, that may have different recognition.
I can show the tree. I don't remember.
Yeah, why don't you show the tree, because I can't remember either. I think what I noticed at that point was that there was a little LIC5 group, and that was what I wrote down. It's always good to
I don't know.
get that.
1 second. I can't. 1 second. I cannot.
If it's hard to share, we can just.
Okay.
There we go. Okay, so let's see here.
So these are the 3 LIC5.
And then you have the circ1.
Yeah, here the circs.
Yeah, but are these the entire collection of lysozyme kinases in Physco?
Of the what?
Lysozyme domain kinase proteins, right?
Is this the full phylogeny for Physcomitrella?
Are there other lysozyme domain proteins there? Receptor-like protein, receptor kinase-like proteins.
It looks like if this is the set, that it just has those 2 groups. That'll be interesting to see whether there's some that are more divergent that wouldn't have come up in this one. Interesting. Okay, let's go back to, why don't you stop sharing and we'll go back to Ronell Roth has a question. Are there fungal pathogens of Physcomitrella? If so, have you tried the LIC5 RNAi lines? for susceptibility. That would be an interesting path.
For other fungal?
Well, just for actual, instead of using purified chitin with an actual fungus.
Oh, we didn't try that, but that would be definitely interesting to do. Yeah.
Yeah, I think that would be a really interesting future direction. And you probably have many people on this who might be interested in collaborating. So remember, if anybody is interested in collaborating or has other questions or contacts, remember that for each paper, the email for the corresponding author is given. So that's another way you can contact after the seminar. Okay. Do you want
next question is from Wei Cui. And I think his question is to ask whether the chitin treatment affects the cell type. So because you have those, in the tissue, you have the chloramina and Nima and colonema cells, right? So
yeah, that's a really good question.
You have these two kinds of cells.
Yeah, this is a really good question. So we, for the actin data that I showed, that was the colonema cell, which we usually always study first because they grow faster. But we didn't specifically look at the response in the colonema cell. It will be definitely interesting to do that. There are some differences in these cells, so it would be interesting to see if the effect is the same. Even the actin cytoskeleton at the tip is bigger, like it's a broader, it's distributed differently. So it would be interesting to look at that. Yeah.
Definitely. Yeah.
Okay. Erbil Gongor asks, you see an increase in expression of PAL chalcone synthase genes. Do you also see, I don't know, ricionidin A or anthocyanidins being produced and causing a red color? Oh, I don't even know.
Yeah, so it's asking whether you have if you observe any secondary metabolites accumulating.
Right.
So
after long-term treatment.
The plants treated with the higher concentration of chitin oligosaccharides have look more brown, like they have callose deposition. We didn't like detect specifically what is the type of like compound, but they are more brown.
So maybe some kind of polyphenolics or something. Yeah, well, that's a whole other area of investigation for people who are interested in metabolomics. What happens in Physcomitrella in response to chitin signals? It's an interesting question.
So next question is from Ravi Kant. So he points out that RNI, of course, can be variable. So he asked whether you have quantified the degree of silencing of the three genes in your line.
If we quantify the degree of silencing?
Yeah, right. How much, are they 100% silenced or 50%?
So this silence mechanism is very efficient, and so in that paper that I referred, we would see up to like 90% of silencing. So yeah, there's a high level of silencing.
And you checked all three genes?
No, I don't remember specifically about that. We checked another gene family and the silencing was about 90%, but I don't remember specifically about the LIG5.
So Francine Govers says, nice presentation. Thank you. I have a question about this novel RNAi method. Do I understand you correctly that you can switch on the silencing and switch it off again and follow recovery of the phenotype? Oh, that's interesting. That you might, I was thinking about it just as selection at the beginning, but that's an interesting idea to be able to remove the selection and switch it off again and follow recovery of the phenotype. Oh, that's a cool idea. So, depending on what you put in the medium.
So when you silence the gene, so the plants that are not silencing, they will die. So it's not like they will be dead. So it's not reversible.
It's not an inducible system, right?
No. Oh, no.
Maybe though, it might be possible to titrate it down so you have just a small amount of that to make them sick. And then, I don't know, that would be an interesting thing. You might be able to play with that.
Well, I don't know. I don't think so.
Mm-hmm. Yeah, that's an interesting question. And she also wonders whether it has been done in higher plants. I think people are excited about your RNAi system here.
I think so. I think that it is translatable.
Mm-hmm.
Yeah.
So Nicola Cecchini asks, Cecchini, how do you compare the frequency of calcium peaks triggered by chitin with other responses?
How do I compare the
or maybe it's not how do you compare it. I think the question is, how does it compare? Like you have your calcium spiking in the tips.
Yeah.
Yeah, and is the frequency there similar to the frequency you see with calcium, with chitin?
Yeah, no, so the frequency is different. And that's, I think we think that in itself, it is something that is signaling the plant. It's a signaling mechanism of the plant because the frequency is different. So that's something that we have to like further study if maybe that can activate a specific kind of like downstream. Yeah.
Yeah, like FM radio. Okay.
So, Paulo Perello asks, apologize first for asking another question, but he says it's such a nice talk. So his question, I think, is which a very interesting one, is that if you treat only a small, at one site, do you see a systemic wave of calcium oscillation or spiking in response to chitin, or some other kind of elicitation, right?
So we didn't. So yeah, this is a very interesting question because we were treating the whole plant and we observed a systemic response. No, we didn't do an experiment. It would be really interesting to do that. That's what happens in Arabidopsis. So yeah, it would be very interesting.
Yeah, right.
Yeah, to sort of treat one part.
Yeah, based on what we see, I think that we will still see a systemic response. The cells are connected with plasmodesmata on the wall between each cell, so they're all connected. So yeah, I think that's
fascinating. Okay, Juan Carlos Diaz Ricci asks, he's asking whether the calcium peaks you observe are due to calcium entering the cytoplasm from outside the cell or the movement of calcium from organelles into the cytoplasm.
It could be both. There is calcium in the medium in which the plants are growing. So
yeah. And when you add the ionophore, it's probably letting it in from the medium.
Yeah.
On the other hand, I'm assuming Physcomitrella has calcium stores in its ER. And vacuole.
And usually those are,
like just like a higher plant. Oh, sorry, not higher plant, just like
no, it's okay.
Just like flowering plants.
Flowering plants, yeah.
Flowering plants, yeah. Okay, all right. I think we're going to stop there. So thanks everyone for a whole bunch of very interesting questions. And Giulia for a very interesting and informative talk. It was a great seminar.
Thank you.
And you handled as many questions as the question period was as long as your seminar. So that's one of the things we try to do. The whole goal of this is to to build community among MPMI members across the world. As you saw at the beginning, people are joining in from everywhere.
Yeah, that was very, very nice.
Yeah, and to foster inclusion. So everybody has a chance to ask a question. So that's always a lot of fun. Giulia did a terrific job as a graduate student managing this international seminar. Very exciting and curious to see what your next steps are.
Thank you.
And if anybody's interested in connecting either with Luis Vidali, you can find his email on the paper, and also can use that as a way to contact Giulia if you have any suggestions or questions for her. So thanks everyone. Our next, that's interesting question. Ashley, what is the date of our next seminar? I think it's in a month, but you can get to that the same place that you registered for this Physcomitrella seminar, you can register for the next one. And she has just typed it into the chat. Thank you. I'm sorry about that background noise. That would be my cat. There's no baby here. Loudest cat in the world. Anyway, so our next one is, where is it? Oh, it is Anne Sicard, who is going to be talking about a very interesting technical advance, looking at inference of bacterial pathogen instantaneous population growth dynamics. And I thought that was a really interesting one. And I think it's nice to have the actual speaker telling you, answering all your questions about a technical advance. Okay, so thanks everyone. Terrific seminar, Giulia, and hope to see you guys back in a month. Remember, if you want to see the recording, you can just go back to the place that you registered.
Thank you. Bye.